If you gave 50 people a piece of paper and told them to fold it however they want, each person would come up with a different shape, whether that's a crane, a frog, or a boat. Proteins work in the same way. They're a very diverse group, and you can create anything from enzymes to catalysts to transporters depending on exactly what sequence of folds you make. All living creatures share the same 21 base amino acids, and the order in which you put those amino acids can completely change how a protein looks. And they don't just fold randomly either, there's preset directions which are encoded in our DNA. This is exactly how origami works as well, because there's only so many folds that you can make, but the exact order in which you do those folds can completely change the final shape. If you're missing even one step in your instructions, then the final origami shape is going to look completely different. It works the same for proteins. Even if there's one DNA mutation, then the final structure is going to look nothing like what your goal is. And this is important because in biology, the structure is the function. If you have a different structure, then it's not going to be able to fulfill its role. Now, if I gave you a piece of paper and gave you all the directions to follow, then everyone would come out with more or less the same shape. This idea that given the sequence of amino acids, you can create the structure of the protein is known as Anfinson's dogma. Now, there's one problem left. How do we go from the actual sequence of amino acids to the 3D structure? If humans tried to randomly guess what every single fold looks like, it would take eons to actually get the real structure. And that's when AI steps in. Because if there's one thing that we know AI is good at, it's taking patterns and learning from them. In 2018, Google DeepMind came out with a software known as AlphaFold that did just that. AlphaFold looks at millions of protein structures and finds patterns like what kinds of amino acids tend to group together, or what kinds of amino acids tend to repel each other. And from this, it is able to predict a protein structure given any sequence of amino acid. This work was so revolutionary that it won the Nobel Prize in Chemistry in 2024. Before AlphaFold, we still had access to all the DNA and access to all the protein sequences, but if we wanted to know what a 3D shape looked like, it would go through expensive lab techniques that could take years. The applications of AlphaFold are far and wide. Scientists can now look at drug structures and protein structures and see how compatible they might be in early stages of testing. We can now sequence thousands of mutations that we know exist in the human genome and see exactly how they affected the structure of proteins, and for diseases, see how they might affect the sensitivity or resistance to certain drugs. We can see how protein structures have changed and evolved across different species and unlock hidden secrets about evolution. Overall, AlphaFold has made huge strides towards solving the protein folding problem. And as members of the scientific community are finding unique and exciting ways to apply this tool, I'll be on the lookout for AlphaFold in the news, and I hope you will be too.